Radboud. I'm today's moderator, and I will be your host today. This is the lineup for the October month. Today we have our first speaker, it's Wilhelm Huck. Next week we have Mohsen Ramani from the Nottingham Trent University, then Laura Fabritz from Polytechnico di Tirano, and then the last speaker of the month will be Kremena Makacheva from the University of Toulouse. This is the whole team today. You may be seeing some familiar faces. So Alice is one of the panelists, Wilfred and Damien too, and they have been speakers before. But let me introduce today you to Wilhelm Huck, professor from the Radboud University. He received his PhD degree in 1997 from David Reinout from the University of Twente, and then moved on to do a postdoc with George Whitesides at Harvard University. After that, he moved to the University of Cambridge and moved back to the Netherlands just a few years later. Since then, he is a professor specialized in supramolecular chemistry with one mission, and that is to understand the complexity and origin of life. He has many awards and grants, but I think one of the biggest he recently landed is a Summit Award for evolving life from non-life molecules. And he got a €97 million Dutch National Growth Initiative. He won a couple of ERCs and FICI grants, and he's also a member of the Royal Netherlands Academy of Arts and Sciences. So let's give a warm welcome to Wilhelm Huck. And Willem, please take over the screen and start your presentation.
Thank you, Chris, for your kind introduction. And I guess you mentioned so much money received that you probably are wondering what I'm doing with all that money. So that's what I will be talking about today then. So it's my great pleasure to be in this iCANX talk. It's the first time that I have the pleasure or the honor to be a speaker. And today we will be talking about neuromorphic computing in chemical reaction networks. And that's quite a long title. And I hope at the end of the hour, you will have an idea of what we are trying to do. So first of all, the background is not chemistry. The background is really that we see an enormous increase in AI applications at the moment. And we all know that the energy that is required to drive all these large language models, to drive your searches for sort of cute little pictures of dogs and cats, is really up eating all the energy that we have. So we need to move towards more energy efficient ways of computing. And one of the ways of doing that is by realizing that we all are sort of carrying a very powerful computer. And that powerful computer is not the phone that you have in your hands maybe right now, but it's the brain inside your skull. So this is an extremely powerful computer, at least for most of us, and it's very, very efficient. So it runs on a sort of minute fraction of the power that a computer chip runs on. So it's only about 20 watts, even though it's the most energy-hungry organ in your body. But for me, as a chemist, I think the most important bit is that this is a molecular computer. So in the end, whatever the brain does, it has to be related to moving molecules around, and especially moving ions around in the synapses that are the connections between the nerves in your brain. And I think this is very interesting, and it has inspired actually people almost from the dawn of computing to think about, can we build a molecular computer? And molecular computers have been, or this idea has been around for a very long time. And the main motivation for this has been that people thought, some people are still thinking, so I copied this text from a recent review, and I thought not to embarrass people, so I didn't put the actual name and the link to the review. But they say the core advantage of molecular computing is its potential to pack vastly more circuitry onto a microchip than silicon will ever be capable of. I don't think so. I think at the moment, if you think about ASML and other Dutch companies making super small transistors, I think maybe the smallest elements in modern computer chips are actually maybe smaller than molecules. But this sort of field was kicked off already sort of 15, 20 years ago. A very nice early example is this work by Fraser Stoddart and James Heath, where they tried to make electrically configurable logic gates out of molecules where you could switch the position of this ring over here between two different positions on this molecular rod-shaped molecule. And so by driving it by an external potential, you could store information in these kind of molecules. Other people have worked on molecular logic gates where really the logic works in solution, and you have molecules that respond or bind to different metal ions, and upon binding the metal ion, either the iron here or the mercury there, you change the color of the overall molecule. And if you combine that in a nice fashion, you get AND gates and OR gates, or maybe slightly more complex Boolean logic gates. That's nice, but really, in a way, it's limited. So if we really try and build computers this way, I think it's going to take an extremely long time. Luckily, other people have made more progress towards molecular computers. One of them is in the audience today. So Chris has worked on artificial synapses, and where he really showed that actually you can make computing elements out of molecules, where these very special molecules sandwiched between two electrodes can really access multiple states, electrochemical states, that mimic the sort of learning and memory storage that you will find in a synapse. So that's an enormous progression towards really encoding Boolean logic in a molecule, but really thinking about different designs on how you can use molecules and incorporate them in electronic devices. Other people have worked on getting away with the electronic devices and work on DNA-based networks. And Chan and Winfrey have made very, very intricate designs of very complex DNA sequences that tumble all over one another and sort of carry out a calculation. Recently, Rondelet and Genot published a paper where they have a combination of DNA and RNA as an input, but using enzymes to actually process that input and build a neuron, or a single neuron that they can concatenate eventually and sort of go towards enzymatic neural networks, if you wish, in a first demonstration. And my former colleague, who passed away, unfortunately, just earlier this year, Roland Nolte, he was dreaming of very different types of molecular information processing devices. He wanted to make a molecular Turing machine where a catalyst runs over a polymer strand and writes information onto that polymer strand via a chemical reaction. Unfortunately, he didn't quite finish that work. Yet, if we think about these kind of approaches, that is not the same as how living systems process information. And the question I would like to ask is, should we really build molecular computers that recreate digital computers? Should we follow that same kind of logic, Boolean logic, to process information? The video that you see here is a slime mold. So it's a single-cell organism that organizes and self-organizes into a multicellular aggregate. And well, it has to search for food. And so if you deposit the different bits and pieces of foods on a sort of virtual lens, on a map, then this colony will start to search for food. And in searching, it will sort of send feedback back to the colony to say, found the food, follow this path, follow this path, follow this path. And essentially in about a day, 24 hours time, it will have actually recreated a network of sort of highway along which the organism can travel that actually resembles the map of the Tokyo Railway network. And so essentially what it has done, it has solved the problem of how to make connections between multiple dots in this network. And so this shows that maybe if we think about how living systems process information, then maybe we can come up with systems that process information, but in a different way. So I think we maybe, well, the advantage of building digital computers, as we know, is that we know how to build them and know how to program them. The disadvantage, especially if you think about the molecular parts of it, the molecular design that is necessary, if you think about the previous slide on the DNA-based networks, the amount of design that goes into designing the sequences becomes, at some point, I think, overwhelming. You have to really pre-program these devices. And in a way that works against the chemistry. We are not allowing to do the chemistry do what it does best, just react with other species and see what it does. And so that also means that these approaches that I've mentioned before, they don't scale very easily because we really have to put in a lot of design for every particular solution that we would like to calculate. And so therefore, we are interested in other paradigms for molecular information processing. And then when we look back at living systems, then I would say this is the most complex molecular computing system we know. And so what you see here is not a wiring of a computer chip. What you see here is essentially the wiring of E. coli, so our humble bacteria that we know so well. And every dot on this map is an enzyme. And every line between the dots is a chemical reaction. And so what you see here is a vast network of enzymes and chemical reactions, but together, this network of chemical reaction allows E. coli to be E. coli. So for those of you who have been studying Stryer or other biochemistry textbooks, remember this is the citric acid cycle, right? So you see it right here. Other bits are fatty acid metabolism or different parts of maybe allowing bacteria to sense glucose in their environment. So essentially all of this is what makes E. coli process information about its environment, take up food, grow, divide, move around, etc. Unfortunately, we don't actually understand precisely how this works, but it tells us that reaction networks of enzymes in this case are super important and super powerful. And so that's why my group is really interested in chemical reaction networks. And we study these chemical reactions networks at many different scales. So we can start with networks on very small molecules, and that is what we will discuss today. So I'll skip this for the moment, but you can also try and rebuild E. coli from the ground up by making small networks of enzymatic reactions. And we have done quite a lot of work on that, trying to make oscillating reaction networks or bistable reaction networks, or build very complex networks and build tools to study them and optimize them, or even maybe do small arithmetic calculations. And finally, we are really interested in understanding how networks in the cell actually function. So we're developing tools to trace the working of cellular signaling networks or genetic networks and see if we can map them and eventually understand the dynamics of these systems, because we don't really know how these vast networks respond to, for example, if you add a drug to a cell, how does that drug impact on the inner workings of a cell? So we are interested in developing sort of tools to study this. But let me go back to this particular system over here, reaction networks of small molecules. So the network that I'm going to talk to you about for the rest of this talk is the so-called foremost reaction network. The foremost reaction network was not invented by us. It exists. It was discovered 150 years ago by Butlov. And essentially what it does, it starts with formaldehyde right here in the middle, and you can react it with glyceraldehyde or maybe dihydroxyacetone, and just mix these in the presence of calcium salt and base, and that's all. So just a little bit of high pH, and then you generate all of these molecules and many, many, many more. You generate hundreds of molecules. And so this is a network that is rather complex, but the reason we choose it is that it is sufficiently complex to exhibit interesting behavior, but also sufficiently small to be able to still study it and make sense of it. And why do I call it sufficiently small? It's because actually when you run this network, what you will find is that there are only a limited set of reactions that can take place. First of all of these reactions between the aldehyde and the small sugar that you see here are aldol addition reactions. And they can be between formaldehyde, But once you make a product, you have another aldehyde. So you can have another aldol addition now between a C2 or C3 or a C4 sugar and another larger compound. So that gives you even larger molecules. So then you build up, build up, build up, but then you have a retroaldol reaction that breaks things down again. And the sort of overall result of this is that every one of these molecules that you see in this scheme can be accessed in every sort of from every other molecule in the network. So I can go from here to there. I can start with formaldehyde and go to there. Any position is possible from any point in the network. So it's a very sort of integrate network, which is recursive because it grows and it sort of collapses. It grows and it collapses. And so this is the network we wish to study. Now, before we get to the computing and information processing part, I need to tell you a little bit more about this network and how we study this. So the first thing we did is that we put this in a so-called flow reactor. And we need a flow reactor because this Formos reaction, if you just let it go and you let it go for half an hour, it will form caramel, sugars. And if you let it go a little bit further, it will form a black tar. And chemists don't like black tar. They cannot really characterize that. So we need a flow reactor, which means that things flush in and out, and then you stop the reaction so it doesn't go to caramel and it doesn't go to tar. So typically 2 to 6 minutes is the residence time. So water goes in, your catalyst goes in, calcium chloride and sodium hydroxide, your formaldehyde, which is the feedstock, and any one of these starting sugars goes in. And then 2 minutes later, things come out again, and then you freeze it, and you have to do a little workup. We don't have to go into detail on this, but then you can inject it in a so-called gas chromatograph coupled to a mass spec. And this allows me to analyze what is going on. It's not so easy, but the first thing you see in the GC is that you see different retention times. So this tells you this is a high molecular weight, not so high molecular weight, lowest molecular weight. Every peak is an individual sugar. Now, in the GC, it's coupled to the mass spec, so you can identify in the mass spec what compound this actually is, and then you know which sugar is actually involved, or at least you can have a proper assignment of the sort of different peaks in here. And so we can quantitatively analyze 51 different sugars, which is not as many as they're made, but the majority of them. And then we're going to test lots of different reaction conditions. We want to understand how does this system deal with information of the environment. And information means I have a different environment. So I have a change in temperature, formaldehyde concentration, catalyst, residence time. Any change in the environment is information. And so we measure all of this, and then we think, okay, we chemists can make sense of that. So what you do is for each experiment, I make a bar chart of every compound that was present. So here are the C6 sugars, the C5, C4, C3, etc. And you do this for lots of different inputs. So every one of these was an experiment. And we try to make sense of this, and it doesn't. So the network responds in a very nonlinear fashion. You cannot say if you change temperature, then this will happen. Something will happen, but I don't know what. So we need to have another way of analyzing this system. So the way we do this is by doing a clustering analysis, where we say, well, for each composition, we look at how much it looks like another composition, essentially. And if you do that, you get a so-called dendrogram, where every one of these pie charts is essentially the bar chart that I showed in the previous slide. So it shows the composition. And the reason why we have a dendrogram is because all of these compositions look more similar to each other than they look to the compositions on another branch. So essentially what this shows you is that the network clustered in response to different inputs in the environment. That's interesting. So at least we don't have an intractable mixture. It looks complex, but it's not intractable. It does something reproducible and in response to a change in the environment. It's just that we don't quite know what. So what we did next is we said, well, what I could do is I could increase the information that I put in. I could, for example, say, instead of just changing the concentration of sodium hydroxide or of a sugar, I could start to modulate the information. So there's additional time information in there. And so what we did here is we modulate the input concentration, and then we look for each of the molecules in the network to see how their concentration changes over time. So you can see it's just like having a rope like in the gym. You're going up and down like this, and the waves travel down the rope down to different molecules. And you see also this amplitude changes as well. So the amplitude is not the same for every molecule, which makes sense because molecules that are connected just via one reaction go up and down with your input. But if they're further away, then things are attenuated, right? Because the wave spreads out across a larger part of the network. So what we did is we developed an algorithm that allows us to say, well, if this is a lower amplitude, then there must be more reactions in between. And we can connect the dots, essentially. So we can say, if this is the input, and there's a certain amplitude, and there is amplitude on each of these molecules that we find in the network, we can construct a pathway and the types of reactions that were necessary to get to that particular point. And then you see that if you had only a little bit of formaldehyde present, then the amplitude, the network is actually rather small. If you have a lot of formaldehyde, the network grows exponentially. So lots of different things become possible, and the sort of input spreads out throughout the network. And so, to cut a long story short, the dendrogram that I've shown you before, where you see the compositions in different areas of the dendrogram, the reason why we have these different compositions is because each of these branches is dominated by a different type of reaction mixture, or types of sort of balance of reactions. So what we show here is that a chemical reaction network responds to the environment by reorganizing. It self-organizes, it becomes a new network. Every condition in the environment generates a new network. That's an important message. So that also means that if you put in a more complex input, so here we put noisy input over time, we get noisy output over time. And we can say, well, how does this noisy input propagate through the network? And we can observe in that case again that this very complex network that we had in the beginning actually contains a central cycle. And from this central cycle, lots of different branches and modules sort of hive off. And depending on your reaction conditions, you follow the blue or the black or the sort of purple arrow and sort of move into a different sort of branch. So the network sort of reorganizes and starts to identify, or you can couple an identification on the network and different input parameters. Okay, that's enough detailed chemistry for many of you, I hope. So what's the take-home message so far? Well, first of all, the chemical reactions in the foremost network, well, as a chemist, I would say they form an algorithm where every molecule can result from or react in any sequence of events. And what kind of sequence of events will happen is determined by the environmental condition. And so this self-organization of the networks, I think, represents a form of information processing. And in the remainder of this talk, I will explain to you how we can exploit this into really a form of unconventional computing. The type of unconventional computing that I want to discuss is kind of a network. And so if you think about a neural network, then typically you have a range of numbers that go in. The network processes these numbers, and you get some number out. Typically, of course, you have to train your neural network to actually make sense and sort of say, well, if you have these numbers, then it must be a 1. Now, that's not really what we have in a chemical reaction network, right? We have molecules going in. Remember, I had formaldehyde, I had starting sugars. Then something happens, and I have molecules coming out. So what we need to do is we need to put this into a more formalized way. And so what we want to do is we want to transfer this into a so-called reservoir computer. And in a reservoir computer, which is a sort of computational framework that has been sort of established some 20, 30 years ago and has been mostly explored in silico, we can actually sort of also do this in a physical system. And what it means is that in a physical system, there's a physical reservoir where agents or parts of the system are connected to one another, forming a highly nonlinear system. Then I have some way of putting information into this system, which is a black box, and something I should be able to measure the state of some of these agents in the system, or something that comes out of the system. And the requirement that people have when you want to build this in silico is that it should be a complex state space. So this output layer should be high dimensional, should not be something one input, one output, because then there is some kind of linear correlation. There should be a lot of nonlinear interactions. Also, what you want is it should be a fading memory effect. What you don't want is that the first input will just keep in there, and then the second input will build on the first one. So at some point, it needs to sort of lose that memory of the previous inputs. And you should have a separation of input signals. So if you have 2 or 3 inputs, they should not all do exactly the same thing, because then there's no linearity. And so what people do in this sort of reservoir, that's a black box. You know the input, you know the output. Typically, it is very difficult in silico to make a very good reservoir. We are not going to bother with that. We outsource this to nature or to chemistry. We only want to look at this readout layer. So what we are going to do is we say, well, we have the molecules going in, we have molecules going out, and we say, well, we put numbers eventually on molecules going in. And when the molecules come out, we need to have a readout layer that allows us to regress this somehow and obtain the correct number. And so how do we do this? Well, first of all, when we started doing this, we realized that the GCMS does not give us enough data. So remember, we can flush things into the CSTR, into the reactor, and then we had to collect it, we had to modify it and inject into a GCMS. What we're doing now is we're no longer modifying anything. We directly inject into the mass spectrometer. And this mass spectrometer is here. It looks very small, but it's taller than me, and I'm not so small. So this is a huge machine, and in goes sort of into the mass spec, and I measure hundreds of ions that are being formed. The rate at which I can collect mass over Z spectra in a mass spectrometer is about 2 Hertz. So if you run this for the whole day and we follow 100 traces, so 100 different ions, we get 15 to 20 million data points. So we collect a lot of data. That's important to keep in mind. How does this look like? Well, here are the pumps that you need to inject the different chemicals. Here is your reactor. It looks like this. So in and out, and you can make it a 200 microliter or 400 microliter or 900 microliters. It's fairly small. Well, for the electronics people in there is huge, but for a chemist, this is fairly small. And then it goes into the mass spectrometer that you see in the background over here. And so we inject it over there. So it goes straight in. And then how do the data look like? Well, this is for an example. So we have the syringe pump, and the syringe pump is pumping in dihydroxyacetone, microliters per minute. And you see this is the flow profile. So it changes every 30 seconds or so. And then this is your traces in the mass spec. So some of them really follow this input profile. Others, they follow a little bit. Others don't follow at all. So the network is just very disordered and very complex to analyze. But this is how the raw data looks like. So then what is the next step we're going to do? Well, we want to build a reservoir computer out of this that can carry out multiple tasks. And I will show you the next 20 minutes or so how we can classify the environment, how we can have a reservoir that understands changes in the environment, and that can even predict the future in chaotic systems. So first of all, how do we do this? Well, I showed you the input on the syringe pump, so with dihydroxyacetone. So that's our flow setup, has a certain input, goes into the reactor, has a certain output. These are the traces in the mass spec. Now, what I need to do next is to say, well, that input is essentially also the input for some target function that I want to calculate. So some function of U over time. And of course, this is my data. So at every time point, I get a list of peaks and the intensity of the peaks. So what I need to do is I need to make sure that during a training period, I say, well, if this is my input and I calculate what the value of the function should be, then this is what the output should be. So the sum of all these peak heights should be this value, which of course it isn't. So I have to apply a set of weights that sort of manages to multiply each of these peaks in such a way that I have the right sum. And of course, in the beginning, that same set of weights for the next time point and the next time point and the next time will be wrong. So I have to minimize the error between the calculated function, the calculated output, and the weights times this output that I have here until I minimize that function so I have the best set of weights. And when I have that, I can now start to predict what the future value of this function will be given a certain value of Wx. And so this is the sort of predict phase. And so what function can I calculate? Really, it doesn't matter. It can just be fu is u squared, can be a differential equation, it can be anything. I'll show you in a minute. So the first thing we did is we said, well, can we do classification? So I go from pH low to high, chemical fuel low to high, and I want to do an XOR gate, Boolean logic. I say, well, all of these are ones, all of these are zero. So I do 100 experiments and I collect the mass spec over time and say, well, this is the average value that comes out of my composition. So at every point in this sort of experiment, every 100 times I get 100 different outputs. And so this output needs to be trained in such a way that this sum of outputs generates a 1. If I take anything in this quadrant here, it would be a 0. And then I find the right set of weights and I can do the classification. Again, mind you, one set of weights to classify all the compositions correctly. Now, this is doable. It's actually extremely good at doing this. So our foremost reservoir computing can do an AND gate, an OR gate, a nonlinear XOR gate, a circular sine wave, any pattern that you want. In principle, even nice pictures of cats and dogs, but then you need more inputs. And so this is one data set. For every one of these sort of objectives, I had a different function. I found a different set of weights. Then you can see here, well, how good is this reservoir? Well, for this particular kind, we can sort of calculate the Matthews correlation coefficient, which is a measure of a score of how good it is. And then you see that it competes on a par, or sometimes even outcompetes machine learning classifiers. So we are sort of compatible, or competitive, I should say, with machine learning, in silico machine learning techniques, even for this very simple task, for which you would say chemistry is not really we are not exploiting the chemistry yet, because this is just a static system, just a fixed input and fixed output. Just give you a bit more insight what happens. So we have these 100 different traces, and if you just follow a few traces, so let's take trace number 19. It's a C6 sugar apparently with calcium, and then you say, well, the intensity of this particular ion across the 100 experiments is measured here. You see some experiments is high, some is low. And then you say, well, during the linear classification, we put weights on there. So we multiplied it by a certain value. Here we sort of fixed these values for all of these different ones. And that gave us an overall contribution to the classification that looks like this, which really doesn't look like the final answer yet, right? So this one trace, yeah, gets these ones right a bit, but got these ones very wrong. And so in each step, we add more ions, traces to the mix. And if you do this and you add many, many more, then you see in this step here, we have a very good classification where most of these fit really well with a -1 and a -1, something in between where they don't fit so well yet. And you see the individual contribution and the individual weights of every one of the ions. So this is just to give you a sort of under the hood picture of what is really happening. Now you could say, as just mentioned, we are just measuring static systems. So we are not measuring dynamics. And living systems are dynamic. A bacteria can sense continuous changing environments. So can we do that as well? Can we mimic how a cell responds to a fluctuating environment? So the way we do that is we said, well, we took the metabolic model of E. coli. So this you can find on the internet. It is about 92 different chemical reactions, 87 different substrates, so 92 differential equations altogether that show you how you go from glucose to all the building blocks that you need in the glycolysis, etc. So then you can calculate the outcome of these 92 differential equations and say, well, okay, if I compare this output with something where I'm running a foremost reservoir, I can do again a training and predict cycle. So how do we do this? Well, this is the actual network. So this is the glycolysis network and the citric acid cycle, as you see here. And we calculate what happens if you have a certain input of the dihydroxyacetone here, how everything in this network would go up and down. And then we do the same thing, and now in a measurement, we take that same fluctuating input, but now the actual input, and we put this into the foremost reaction. So we don't have this metabolic network in our reactor, we have the foremost. So this is our input trace, this is our output. Now, then you say, well, I can look at my calculation. So the red line is when I solve the differential equations, and I get concentrations for pyruvate in time, 3Pg, adenosine monophosphate. So sorry, what dropped off here is minutes. So it's changing in time. So the orange trace, what we have here, is the training phase. So for 30 minutes, I train my reservoir to predict the continuous change in concentration of pyruvate. And then you see, once you've found a set of weights, it predicts the concentration of pyruvate rather accurately. You can do the same thing for 3Pg, same thing for AMP. Actually, you can do this for all 82 intermediates in the E. coli core metabolism, and you see almost all of them, whether it's a millimolar or a micromolar concentration, your foremost reservoir can fit essentially the outcome of all of these differential equations. So this tells you how powerful this foremost reservoir actually is. Can we do more? Well, the more exciting bit would now be if you say, can we predict the future? And you would say, we can't predict the future, but actually your brain can predict the future. This little boy knows where this ball is going to be. So his hand is already there to make sure that it is going to catch the ball. So you can predict where this ball will be in the future. Your brain predicts where your arm will have to be. And you can do this for a little boy catching a baseball. It works all over the world. You can also predict the ball and hit it with a ping pong bat. So your brain can predict the future. What about the foremost reaction? Well, that means that if I have some kind of chaotic input where I'm sort of changing the input here, I'm learning what the output is if I have this particular input. So I'm trying to learn this function. Then when I say we reach this point over here, now I have measured this particular combination of output signals. What I need to predict is what will be the input in the future to continue apparently along this particular input trajectory. So my reservoir, my output here needs to predict the input that I'm going to give. So this sounds rather spooky, but actually it works. So if you change the concentration, so you have a certain sort of pattern in mind and you train it on using the dihydroxy acid input, sodium hydroxide input, or the formaldehyde input, and I follow this sort of oscillating pattern, then indeed I'm able to predict the future a few steps ahead or one step ahead. This works well for the dihydroxyacetone. Sodium hydroxide works even better. You also see if you have formaldehyde going into the network, apparently your network is not so sensitive. Apparently that is a sort of part of the regime where the self-organization of the foremost doesn't lead to something that is apparently a very good reservoir computer. So that's actually quite interesting because it means that this self-organizing reservoir computer is not always equally good depending on what you use as a fluctuating input. So we can have a look at that a little bit more and we say, well, what if we have this particular change in input and then look at each trace in our network and say, well, how does the information that we sort of put into the network actually travel through the network? And so we can do that by calculating the mutual information, which tells us how much information propagates from input to output. So you see this particular ion here has a very high score. Apparently the input and the output for this particular ion correlate very strongly, but it drops off for other species. So there's also species where the species are fluctuating in time, but they apparently are just doing things totally oblivious almost to what the input is actually doing. So it has dissipated so far. Think about the ropes. There were so many ropes connected to that first rope that your molecule here is on a side branch and doesn't really know what's going on in the rest. So we can analyze that a bit more. And we can say, well, if we then think about this forecasting and memory, then you will find that different traces and also different inputs. So the formaldehyde here is doing really badly. Has very little memory and also little forecasting capability. So on the left of 0 is memory, on the right is forecasting. And then you see that, especially for sodium hydroxide and DHA, you see there's quite a few ions, that ion traces, that seem to have a memory of previous events in the network. So these are the ones that apparently keep up with, well, there's lingering in this network. You can also see ions that really are much better in almost forecasting the future. So they pick up another dynamic in this system. So all of these ions respond in a condition-dependent and timescale-dependent way. And so this is exciting, I think, because we're really learning about how chemical reaction networks process information. And it's opening up a whole range of new experiments that you can think of. Well, before we get to that question, I think I would like to sort of also ask the audience, what should we be thinking of? A little bit more, what are we doing? Well, so far we have a computer that needs to set the weight. So we have the mass spec, measures hundreds of different ions, and we need the ion traces loaded into a computer, and the computer has to do the regression and sets weights on each of the ions, and then we can do the classification. But if you think about bacteria, it doesn't really have to measure something and send it to a computer. Your brain, to catch the ball, was capable of doing this by itself. So can we go towards a fully chemical computer that really senses the information, processes it, and sort of define a response? So can we design a chemical classification system? Not yet, but we have made a little start. And we realized that when you have the reaction mixture, then of course we measure a range of different ions in the mass spec, but what you actually have is a complex chemical mixture of aldehydes and ketones, right? These are all the different sugars. And it turns out that there are many reagents out there, it's Benedict's reagents or Silivanov's, that are colorimetric assays to sort of determine the presence or absence of, in this case, ketose or aldose, right? People are interested in this. They want to know, do we have ketose sugars in there or not? And you change the color from sort of pinkish to deep red. You have another Benedict's reagents is with a metal ion. There's copper in there. So you start with a blue solution. If you have some reducing sugar, it goes a bit to yellow-green. But if you have a large amount of reducing sugar, it goes to red. So the idea is that for every different input combination, we get a different output combination. We know that. And we thought that this output contains a wide range of compounds, right? Not just one compound, yes or no. But we thought if we add a reagent like this, every time you add a reagent, it will react slightly different with this complex mixture that comes out of the formose. And maybe that gives us a slightly different outcome that we can measure. And so then you could think about classification where you say, well, I had different DHA concentration, different sodium hydroxide concentration. For each of these reactions, I have a different output. Now, I add my reagent, one reagent, but this reagent has a slightly different reactivity for every of the compounds in there. So if you then wait a little bit or I heat, every one of these compounds gives me a slightly different colored compound. And in total, that gives me one color output. And indeed, this is what you see here. So if you then just take an image on your mobile phone, then you see that now the formose reaction has self-classified. It has sort of identified the different conditions. So the differences are small, but I can really do a classification based on looking at the color, or maybe I can use different reagents. These are examples of different reagents, clearly giving you a different shade or a different intensity. Now, I appreciate we are not at the moment learning how to optimize this, so I cannot find the best set of weights yet. But it's the first step towards a chemical readout rather than a computer readout. Can we do something else? Well, the other thing is that at the moment, every time we have to program the syringes, they go to the reactor, they go to the mass spec, we measure the data, we do classification. That's it, afterwards. It would be nice if we could start the syringes, go to the reactor, go to the mass spec, extract the data, and immediately do the classification and send a new signal to the syringes. So then we would have a self-driving chemical brain, essentially, which would run autonomously. And we thought it would be interesting to see if this chemical brain can find its optimal target composition, or could it respond to changes in the environment on the fly? We've made some progress with that. So what we are doing at the moment, where we say, okay, we can again change the flow rate of dihydroxyacetone. We can change the flow rate of calcium chloride. And then we measure, suppose we are some initial state here, and that gives you a certain composition. But we know that we want to have this as a target. Maybe we have been there before, maybe we classify things into ones and zeros before, so we want to have an ideal pattern again. We want to make sure that it reaches this target. So it should have this composition here. And so the objective now is to move around this landscape of different inputs until it really matches something that is close to this. And so this would essentially be a chemical system that would become robust, right? Because suppose this is where you want to be, suppose there's a change in the environment and you drift off and your flow rate is off, then you still need to find autonomously, you need to find your way back again. Now, there are different algorithms for this, and one of the algorithms that turned out to work really well is the Bateson-Hoppen algorithm. That is a optimization technique that sort of makes a random change, and then it sort of, you can locally sort of say, okay, does my change result into a sort of better match to where I want to be? And if not, choose a random new direction. And what you see here is sort of a couple of runs where we start here, and then we just move around, and the Bateson-Hoppen algorithm sort of tries to get close, and then you see here, it gets reasonably close. And so this is the first step towards a sort of system that can always find back its starting or its optimum position. Mind you, what you also see is that it doesn't get quite to the target value. And the reason for that is that if you think about this chemical output, then at every possible combination of input conditions, it gives it a slightly different output. But this output difference can become really small, right? Because you have 100 different ion traces. And after a while, this very small difference will give you a very small difference in output. But this difference might be so small that the algorithm thinks it is close enough. And that is why it doesn't quite hit the spot. It gets sort of stuck in an environment where you say, well, all of this is probably close enough. The other system that we have been trying to work on is to say, well, can this system become a self-driving system, essentially? So could we do something where you say, I have an oscillating input of sodium hydroxide, I have an oscillating input of dihydroxyacetone, I train the algorithm to recognize that I have this particular oscillating input, and I say, this is what you should stick to. So the dotted red line is this continuous input fluctuation that I would like to keep. So then, of course, it has learned that it is oscillating. And so after, say, 2 hours, we say, you have learned enough, now go ahead. And so you just do one step ahead prediction. So the blue traces that you see is the actual input concentrate, or the flow rates that are suggested now by my reservoir computer to ensure that I keep oscillating. And, well, I think you see a couple of interesting things. So clearly, it doesn't work. So that's one interesting observation. Your reservoir computer is able to predict the future one or two steps ahead, or a few steps ahead, a few seconds ahead. And it should match on to this oscillating system. And it does keep oscillating, as you can see. So it has learned something, but for some reason, it seems to show interference patterns, right? It shows some interesting additional frequencies. You can see here, additional frequencies are emerging, also on this one. So what apparently is happening inside the reservoir computer, inside this complex network, apparently it generates additional frequencies or additional patterns in there. And so I think that makes at the moment means that our reservoir cannot quite keep the amplitude and the frequency of the dynamic inputs sort of correct. But I think it shows us something else where maybe this network is trying to do more. Maybe it's generating new signals, and maybe we can learn from that. Maybe in the future we can learn what this network is actually doing and say, well, is there some intrinsic property of a chemical reaction network that if you feed it with two different waves, all kinds of new frequencies start to arise and emerge, and maybe we can tap into that and learn something more what the network is doing. So at the moment, this is work in progress. I hope in about half a year's time, I hope I can show you that indeed we can train this network fully, and maybe it can play a computer game with you and see who is a better player of simple computer games. So that's Thijs de Jong's project. Hopefully we'll get there soon. Another area that we want to go is, you would say, well, I mentioned at the beginning that we were inspired by AI and computing. And then you would say, well, you've drifted off into complex chemical reaction networks, and you have this reactor and you have a huge mass spectrometer, and it looks dirty and says it smells of caramel after a while. That doesn't sound like a real computer. Do you want to have a real chemical reactor and a mass spec on your desktop to compute something? I don't know. But in a European project called CoreNet, in collaboration with other partners, especially IBM Research in Zurich, they're actually quite keen to see how far we can miniaturize and integrate things. So you could think of a chemical reactor that is really CMOS compatible, really integrated into a monolithic chip where everything is controlled by electronics. You have little valves where reagents go in, where all the different sort of external control parameters can be programmed. And then the outlet is not some dripping reactor or some syringe that goes into a mass spectrometer, but really a channel on a chip that you can measure with FTIR or UV-Vis, or maybe some sample extraction, etc. But sort of there are different ways to measure these compounds. So we have chosen mass spectrometry, but you could imagine that for different types of species that you want to pick up, maybe FTIR or UV-Vis will be possible. And so, well, I think it's IBM's vision to say, well, how far can we push this? Could we really think of building a chip that contains a chemical computer? And if everything is closed, then, well, maybe this is not so dangerous as you think it might be. So this is exciting, and again, something that is being developed at the moment. Now, where do we stand at the moment, and where do we want to go? I've shown you that the Formos reaction is, I think, an extremely powerful reservoir computer. It is on a par with in silico machine learning algorithms that people use all the time. So I think it's definitely working. And I think we can improve our computational power by parallelization, or making deep reservoirs where we can have multiple reactors in series or maybe in parallel, and maybe that would even make things more powerful. I think also we can go to a self-driving reservoir computer where we really sort of tap the data directly from our measurement analytical tool and use it to pump in new values for the pumps. So also that will be sort of something that is feasible. The question is, where do we want to go? Should we develop a chemical brain for a soft robot? Would that be the way to go? Because this chemical brain can respond to chemical changes in the environment. So a computer doesn't measure pH or salt or sugar concentration. Our Formos reaction, of course, does that, right? It senses chemistry. Or should we make it into a sensor, maybe for artificial skin or something, and have really, again, this sensing capabilities, not only physical, but also chemical, or maybe even smell? Or should we go towards more computational power and really sort of build alpha Formos, have a supercomputer made out of molecules? I don't know. Should talk to Lee Cronin maybe here. So the question I have for the audience, what should molecular computers really compute? What are the best things that we should be doing, assuming that a chemical computer is actually really powerful? So don't hold back. And the other thing is, what we do need to make sure of is that we exploit the unique features of chemistry. And I think I've shown you that we already are capable of doing that, because in the example where I showed you the E. coli core metabolism, we really exploit the fact that we have dynamics, right? So chemical reactions have this intrinsic dynamics because of the different timescales of chemical reactions. And that allows us to compute these the differential equations that are part of the E. coli metabolism, because they have similar timescales involved, similar dynamics. We also saw in this last example that I showed you, where we're putting two oscillating input patterns, that we generate more input sort of oscillations, I think, in the system. So these are unique features of chemistry that really responds where every chemical reaction is dependent on the concentration of each species at that particular time point in the reactor. And that is something that really is very different. So our network is not fixed. So if you think about an in silico reservoir computer or something that is fixed in an electronic device, it's fixed. It cannot just reorganize. But a chemical reaction network reorganizes all the time. It can be any reaction network that you want it to be. That is sometimes really bad, but I think we also should be thinking about how we can exploit that unique feature of chemistry. So with that, I've come to the end of my presentation. I look forward to ideas that maybe start coming up. I want to thank a couple of people. I would like to thank, first of all, Mathieu Baltussen, who has been really the star, who has done all the calculation work. So he has done all the formal work on the reservoir computing to really sort of develop the training and the testing bit of the sort of stationary phases, the dynamic, the molecular, the sort of the forecasting part, etc. Then I want to thank Thijs because, well, Mathieu needs experimental data. And without Thijs, we wouldn't have any of the experimental data that is in the reservoir computing work. So he has been brilliant in working that. And he did this together with Quentin, who's a postdoc who's now in Belgium, who is the mass spec expert. And without a mass spec expert, this work would not have been possible. That said, Will and Pierre and Lena and Thijs worked also on the characterization of the foremost reaction. So the earlier work published in Nature Chemistry and JAX, where we really sort of did a lot of work to really identify and characterize and familiarize ourselves with all aspects of this foremost reaction, to think about how we should think and map sort of something that was so complex, and really gave us all the sort of starting sort of material for thinking about these kind of systems. That first work was sponsored by the Simons Foundation, the Radboud University, and ERC Advanced Grant, and all the work on the reservoir computing is again funded by the ERC Advanced Grant and the CoreNet consortium that I mentioned before. Of course, there are other people in the group whose work I wasn't able to touch on today. There are many people working on chemistry and AI and reaction networks, but that's hopefully for another occasion. And with that, I would like to thank you all for your kind attention, and I look forward to the discussion. Thank you.
Thank you, Willem, for an excellent talk. And now let me share my screen again. Let's move on to the panel discussion, and let me now introduce all panelists. Today we have Alice on board, and I think everybody knows Alice already, so he really doesn't require much of an introduction. She's a professor at University, but I think we all know her very well because she has been the founder of the International University Students iCANX Innovation and Entrepreneurship Competition. She's the chairman of the International iCANX Alliance and the Secretary General of the Global Chinese Micro Nano Molecular Systems Society. Then we have today also Wilfried van der Wiel, and he is my dear colleague at the University of Twente. He is a professor at Nanoelectronics and the director of the Brain Center for Brain-Inspired Nanosystems. He holds a second professorship at the University of Münster, and his research focuses on unconventional electronics for efficient information processing. And then we have Damien Thompson, and I'm also very delighted to introduce him because we have been collaborating for about 15 years, and it has always been a pleasure to work together with him. He is a professor at the University of Limerick, and he focuses on miniaturization and sustainability. He is the scientific director of the Science Foundation of Ireland Research Centre for Biopharmaceuticals and leads the modeling activities of the Centre in Rational Design of Novel Supramolecular Structures. His research interests span from quantum mechanical modeling to mesoscale modeling. And sometimes I know for a fact he also does a little bit of DFT. So welcome you all. And let's now start with the panel discussion. And maybe I can invite Wilfred to kick off.
Yeah, thanks, Chris, for the introduction. And most of all, thanks, Wilhelm, for a really fascinating talk. Really a very comprehensive overview of your activities on these chemical reaction networks. It's really nice to see how you brought apparently very remote fields together from this very fundamental chemistry to classification, reservoir computing, and this is really wonderful to see that you merge those fields. I had a couple of questions to you. Maybe I'll just fire them and then you can decide whether to ignore part of them or spend more time on one or the other. So you mentioned correctly that this concept of reservoir computing was actually born as a software model, basically, as an approach to solve issues with recurrent neural networks in modeling. But now over the years there have been quite I could even call it a zoo of different proposals for physical reservoirs, right? And they really go, they range from a swimming pool to all kind of exotic systems, electronic, magnetic, mechanical. So I was wondering, where do you really see, in the context of physical reservoirs, where do you really see the edge, the great advantage of your system? And of course, accuracy is one important figure of merit, but there are other figures of merit as well. So maybe you could comment on that in a moment. I had another question. So I really liked your direction to get rid of the external computer. So Herbert Jäger was basically godfather of the reservoir computing. He always tells me, "Oh, we have to throw this digital computer out of the window because this is really nagging." So you're going into that direction. But I was thinking while you were talking, which is not optimal, but if you really would like to design a preset mixture, like basically nature also does, we have a kind of somehow learned a golden mixture, or at least good enough for purpose, and then it does the job that it has to do. So could you think of a way that you can already pre-design a mixture of all kind of components that is fit for the job, and how would you envision such a thing? So that's another question. My third question was what you often hear in the context of reservoir computing is that it seems to be advantageous to operate at the so-called edge of chaos. This is a kind of popular slogan. So I was just wondering how far away, or do you have sometimes chaotic systems? Do your networks always produce reproducible behavior, or somehow you can also get into a situation that there is basically nothing to begin with your whole mixture? And then last but not least is in your introduction you gave one of your main drives in science in general is the origin of life, which I find really fascinating. So how does this work connect to that, and have you been able to learn something from all of this for addressing this truly grandiose question of the origin of life? So with that, I remain silent. Yeah.
Thank you, Wilfred. That's a lot of questions, and I think.
Can I interrupt you, Willem, for a quick second?
Yes.
Wilfred has indeed a long list of questions. I hope you wrote them all down.
Yes.
But I hope this will become a lively discussion. So also the other panel members, please jump in if you feel like, okay? Please continue, Willem, sorry.
Okay. Yeah, the edge of chaos. I personally feel that I'm constantly at the edge of chaos, but chemistry is not. So these are really deterministic systems. Although they are very complex, they are perfectly reproducible. So we're definitely not there. Then thinking about competition with physical reservoirs, you could say, well, swapping one physical system for another, whether it's a bucket of water or quantum dots or chemical reactions, I agree with you at a certain level. That doesn't really add anything to the very concept. You could say, I think the chemical reservoir has the advantage of having inherent dynamics that are sort of suitable for calculating other systems with similar kind of dynamics. So I think if you want to look at biological systems or maybe even climate, then a chemical reservoir has maybe certain advantages over one that is electronic and where everything happens on a sort of a billion times faster time scale. But other than that, I don't 100% sure. I feel we have to move towards getting rid of the digital computer part. And that is where I think chemistry could do a good job, because there are different ways. So I think if you think about the Formers reaction, then what people have done is actually they have interacted the Formers reaction with cells. And so it turns out that cells were able to grow or not grow on the mixture that is produced by cells. So you could say, well, I could maybe train cells to recognize certain mixtures and not other mixtures. So that would be one direction. In another area where I've not discussed today, but we have another physical reservoir or chemical reservoir which is based on enzymes. And there the output will be peptides. And so rather than sugar molecules. And again, peptides, you could say, well, in both cases, either the sugars or the peptides, if you want to do a physical learning, then I think what you should do is to say, I take a number of different components. So think about the reagents that we had earlier. Do not come up with one particular mixture which gives a certain output, but allow yourself to combine sort of maybe 6 or 7 different parts of the reagents in such a way that you train that kind of mixture of reagents in such a way to do the classification. And then I think you have learned, so that the system has learned how to classify one particular sort of environment. So it senses maybe high or low pH, and it gives you yes or no answer exactly when the pH is above or below that particular value. So of course, then you cannot do anything else, right? Because in the computer, it's very easy to take the same data and find a new set of weights. If you do the physical learning, then the physical system will learn to classify for that particular yes or no answer, but it cannot really adapt afterwards. But I think that also means that, again, we have to think about where we are going to use this. And I thought, well, maybe chemistry goes towards biology. Can we think of something medical or sensing in the body and give an output that is yes or no, but really not a yes or no output on a screen, but a yes or no output in terms of switching on or off a gene, for example, something in that direction ultimately. But honestly, we're not even close to that. And then to finally touch on your question with the origin of life, I'm very excited about this. I don't think that a reservoir computer is the origin of life, but definitely information processing is at the very heart of what living systems do. And I hope we can use this insight to build on reaction networks that have the power of evolution, so they can take on board more complex reactions. And maybe we learn how to then do some kind of selection mechanism. So suppose your Formers reaction would be exposed to metal ions, and then it generates sugars that become ligands for these metal ions. And when they form a complex between the metal ions and the ligands, then they become a catalyst for a certain part of the chemical reaction network to drive it ever further from the starting position you were. And that feels like a sort of taking on board information and evolving into something more complex, which is still far away from the origin of life, I'm afraid. But that's a sort of probably question that can keep us all busy for quite a while to come.
Yeah, great. Fascinating. Thanks a lot, Willem.
Yeah, thanks a lot. regarding your answer that your system is not chaotic, that surprises me a little bit, because many chaotic systems are very simple, even 2 or 3 differential equations that are coupled already produce chaotic systems. You have far more coupled differential equations. I would guess you can find chaotic regimes.
Okay, that might be. Maybe we didn't search specifically for that, but if you recall the example where we had 100 different experiments with different sodium hydroxide and say dihydroxyacetone concentration, if we revisit one of those experiments after doing 100 other ones, we get exactly the same composition, exactly within sort of being able to put the same weights and get the same classification. So it is remarkably reproducible. And I think that's because chemistry on that scale, you have really a lot of molecules, right? So there might be the odd molecule that is doing something stochastic, but really it is driven by a set of differential equations where there's nothing really in a stochastic regime. So we were thinking about maybe making the reactors smaller and smaller and smaller, then maybe you get more into that regime. But at the moment, we don't find that.
Yeah, okay. But the last results we showed, these oscillations, yeah, they were maybe a little bit stable, maybe a little bit not unstable, but then they recovered again. You could also do some analysis on that. Maybe there is already some edge of chaos hidden in there. Phase portrait plots, something like that, I think should reveal that.
Yeah, we are working on that, and really we should have Mathieu Baltus himself in the Zoom. So he's doing something called dynamic mode decomposition to really look at the different aspects that are present in the network, and we do find all kinds of different patterns. So not everything oscillates actually. So if you look at different species, then some of them have a really strange pattern for a little while and then do nothing and then restart again. So there's all kinds of modes in the network that we didn't expect. What I don't know at the moment is what it means or whether I can use it or whether it's interesting or something. It's really sort of we're exploring at the moment.
Yeah, sounds very interesting to me, actually. Well, thank you. Yeah. Wilfred, are you happy with the answers?
Absolutely, yeah. No, I realized they were not the easiest. So, but yeah, I mean, I'm really fascinated by this work. So I like it a lot. Yeah.
Okay, then maybe we can move on to the next panelist. Damien, would you like to continue? Damien, you are muted, I think. I cannot hear Damien. Can you hear Damien?
Okay. Oh, okay. Yeah. David, can you be a little bit closer to your microphone?
Can you hear me now?
Yeah, but very, very silently, not clearly.
Okay, that's weird. I'm not sure why that is.
Okay, maybe I jump in. Okay.
Yeah.
David, you work on your microphone. I jump in for the questions. Okay. Yeah.
Okay.
So really, yeah, really exciting, very inspiring talk. So yeah. So I just have a quick question for the prediction. So it's quite amazing, for that part we see that follow so well. So you see you use the like the readout, as input, right? Yeah, it's like feedback. Does this will affect the next generation prediction? Well, this because you use, yeah, like this was a decision, right? Yeah, get involved, right? So that's my question.
Yeah, so it's a little bit tricky, right? Because think also yourself when you're walking down the stairs, your brain knows where the stairs are because your legs are already moving in the right direction. And if sometimes you miscalculate and there's an extra step, you fall down the stairs, right? So it also sort of works a little bit like that in the chemical reservoir. So if you have too many steps ahead, the system loses track, so it cannot do that. So what you need to do is you train it for a certain amount of time, and you really get a very good set of weights that really capture that movement that you put in and the output that you get out. And then you can do a sort of 30-second or 1 minute or 2 minute ahead prediction. and you just try out how far you can go before you just lose it. Because remember, our reactor has a certain refresh rate, right? So everything flushes in, flushes out. So the average time in the reactor is only 2 to 4 minutes, depending on the flow rate. So obviously you cannot predict 10 minutes ahead because there is no memory on something that was sort of occurring 10 minutes in the past. So there is a window, and it depends on the exact conditions. But it is really sort of something that you have to play around with. And then what we were actually very surprised to see is that it also depends on the type of input that you give, because sodium hydroxide apparently makes the network so much more diverse than when you add formaldehyde, that with formaldehyde, it doesn't really work very well. And the reason for that is if you think about the reaction network, formaldehyde just rushes ahead, right? It just adds carbons, add carbons, add carbons. So, but it doesn't really then make things more diverse. It just makes everything a little bit bigger. If you think about sodium hydroxide, catalyzes every reaction in the network. It catalyzes the aldol condensation, the proton transfer, the retroaldol condensation. So it means that if you add more sodium hydroxide, you simply make many more different compounds. And apparently that is what you, in this particular instance, need for really following the input-output correlation. So I think it's something that we need to explore more in the future to really sort of also investigate how networks respond.
Okay, great. I think there are a lot of things, to be understand. Yeah. Okay, David, you come in.
Okay, so can I try again? Can you hear me now?
Yep.
Yes,
Yeah.
loud and clearly.
Great, thank you. Phew. So yeah, thank you for a great talk. I enjoyed every part of it, and I'm interested in everything you do, actually. So I have a few questions, and particularly I like the idea of the interface with biology and trying to learn something from these systems about how complex life or even rudimentary life evolves. So I'm just wondering about, say, I know you have positive and negative loop feedback loops in your system, and you engineer them into the system. Would it be silly to try to make analogies then between what you learned there to things like, say, tRNA acetylation, which involves a lot of kinetic proofreading, and there's all these kinds of error checks that have evolved in biology. So can you make time models of those? Maybe we could learn a little bit more about how our proteins have actually managed to evolve such efficiency and such such error minimization.
That's super difficult question. Let's see. I think at the moment, well, I think your question is super interesting, but it also tells us that we should explore broader chemistries, because at the moment we only have 5 reactions. So one problem is that these 5 reactions are always on. They're just going. And so what you would like then, if you think about the biology, you separate timescales in a way, right? Because you have your proofreading and your transcription translation going on, but your information is stored in a much more stable position, and the output, the protein, is also a much more stable position. So there's a separation of timescales. And I think in our system, that separation is not big enough. You would like to have, maybe by introducing additional chemistries, something that would really become a molecule that would be much less reactive, or something that would reside for a longer time. And then I agree with you, then you should have a system where you say, well, select only for those molecules that then are the correct ones. So some error correcting mechanism that says, well, under these conditions, these molecules become a signature of that condition or something. But at the moment, haven't got anything yet, because this network responds immediately to a change, and it will just reorganize again.
Okay, no, I agree with you. I mean, it's the first step towards.
Your sound is off again.
Oh no! Can you hear now?
Barely.
Sorry, I don't know what's wrong.
No, it is good.
Oh yeah, okay. I'll stay right here. So yeah, no, I appreciate they're difficult questions, and these are first steps towards very large questions. Yeah, so I wonder, can you turn on your head? So at the moment you have your, your foremost reaction, and you get the electrical signature from that. Can you turn it on? Turn it on its head. Can you use an unknown reaction, and can you track it and somehow interpret what the reaction pathway may be from the electrical output? Fingerprinting essentially reaction pathways.
Interesting. So you say, can the foremost reaction learn unknown chemistry, essentially, right? Say,
precisely, yes.
That's very interesting. I think at the moment we can't. but it's a very interesting idea that you say, can I use a chemical reservoir to learn about chemical reactions?
Yes.
Yeah, very interesting. I don't know how to do that immediately, but you could imagine that you say, suppose I train my reservoir under a set of conditions, and then maybe add a new reagent that introduces a different chemical reaction. So you could think of adding a thiol or a cyanide, which will react with the aldehydes to form a hemithioacetal or something. So that would be a new chemical reaction. And then you would say, okay, if I compare those two signatures, will the system learn what is different or something, right? And then identify what that is, or maybe learn new kinetics of a chemical reaction, have a chemical trace in there, a dynamic in there. It's a very interesting idea for a new direction where you would use a chemical reservoir.
I think you already gave a very nice example of that, actually, where you talked about, say, formation of maybe metal organic complexes inside the body. So you could maybe you could make, you know, DNA damaging agents inside the body. You could recruit, say, biological ions like coppers or magnesiums into complexes that could then be used to damage DNA, say, of microbes or bacteria. So I think, yeah, there's the possibilities are endless. But I mean, we have a fully functioning, worked out example of radical nanotechnology. That's ourselves. So life is there. We know it works. We just have to figure out how to do it. Right? Yeah, I disagree with you, actually. I respectfully disagree with you that molecular design is an obstacle. I don't think it is anymore. I think we have the modeling tools now, especially with AI, where we can absolutely go wild in terms of designing. I mean, in terms of chemical space, of synthetics, in terms of biomolecules. I mean, biomolecules are just what evolutionary stumbled upon. So I think we can absolutely explore a vast amount of chemical space. I don't think that's an obstacle anymore. But if I could ask you one, sorry.
You would still have to make those molecules at some point.
Oh yeah, but I know. They all look fine on my computer, and that's the fun stuff. But again, it will keep us all in work for the rest of our lives, and probably our children, our children's children too, which is great. The final question I had was in terms of the computing ability. So, for example, you said you use these for Boolean logic. How reliable are they? Say, if you measure by truth tables. What kind of fidelity can they achieve compared to, say, memristors or the other molecular examples like molecular tunnel junctions for these types of Boolean logic or molecular logic applications? How reliable are they?
I wouldn't know at the top of my head, but I would imagine that for just Boolean logic, performance is super strong, but so are all the other algorithms that you use. So you really want to challenge it with more complex tasks, which is the set of ODEs. But then I think I need to talk to Wilfred probably to say, well, how do you now classify which reservoir is the strongest reservoir? So at the moment, what people in a way do is just empirical. You just say, here's the same task, just see which one has the largest error, residual error or something. That would be one way of doing it. We would not be so good on, say, well, the Boolean logic is easy, but if you say typical tasks that people often have is this handwriting database or the MS&T database, we would be very bad at that because encoding that into changes in the chemical environment is tedious. So we would rather have time series than sort of spatial sort of tasks. But then it becomes, I don't know if there's a formal way to say, well, here is memristor reservoir, here's an in silico, here is a quantum dot, and here is a chemical reaction, and say, how can you compare them in such a way that I'm not comparing apples and pears in a formal way? I don't really know how to do that.
But I think that so I think what you've developed, I mean, it's liquid, and ultimately it can actually be electrical free in terms of both inputs and outputs. That's wild. I mean, in terms of the interfacing with biology, that's super fascinating, I think. Thank you very much. I really enjoyed speaking with you.
Thank you.
Yeah, if I just may add about the last thing, I think there are so many problems that need to be solved. I think for different problem sets, you will definitely need different solutions for that, different material systems, different chemical systems. I don't think there will be just one winner takes it all like silicon did in the past. So I think we need very different things, and it is very nice to see this complementary thing. But Wilfred, you also wanted to say something?
Yeah, very briefly. So about this kind of generic metrics to compare different reservoirs. So there are some people who really have spent time on this to develop such metrics like memory capacity, kernel quality, these kind of things, and more advanced derivatives of that. So maybe you have to look at those kind of metrics and to make a fair comparison, as you
Yeah,
correctly say,
yeah.
between the two, as an alternative to benchmark tasks, basically.
Mhm.
No, that's just a brief interjection. Yeah.
And then there's maybe one question I had for myself, Willem. Yeah, we make it very difficult for you, very hard for you to earn your certificate today, yeah? But you mentioned in the beginning that at the start of your talk, but one of the major justifications for doing neuromorphics in general is the energy benefit, right? Like you, I'm also very, very much intrigued by our molecular computer between our ears that is incredibly energy efficient. So do you have an idea of the energy efficiency of your system?
That's a really mean question, right? Because you know that huge mass spectrometer uses a lot of
Yeah.
energy. So no, that is why a couple of things. I mentioned the enzymatic reservoir a little bit earlier, which we are prototyping at the moment, which is a mixture of enzymes and a mixture of peptides. And so mixture of enzymes of proteases. So if you add peptides, they're all chopped up. And so you get a similar kind of idea. Everything can be chopped up by everything, can be an inhibitor for everything, a competitive substrate, etc. And there the advantage is that the enzymes you could just immobilize in a material or something. So, and your peptides just come out and then maybe have an effect. So I think we can only become energy efficient if we get really rid of the the electronic readout and the computer as a readout, because otherwise we will be stuck with some huge machine that measures things. Well, maybe an infrared would be okay, but we have to get rid of the computer in between. So we want to move towards a fully freestanding chemical reservoir that can is pre-trained to do classification on something at least. So we're working towards that, because otherwise I think I don't think I can deliver on this energy efficient computing part very easily.
Yeah, but then if we would just zoom in on the network itself, so forget for the moment about the readout path that you are now trying really to work on. The network itself, they're just chemical reactions, so that should be quite energy efficient.
Very chemical efficient, because it's all exothermic reactions, right? So it doesn't require me pumping in heat. So the chemicals are fine. But then you could say, yeah, but somebody had to produce those chemicals, right? So we don't have that in the equation. So, but the system itself doesn't cost energy.
Yeah, and what about a potential or a pathway to electrical readouts? Would it be interesting to study redox active networks, so you have a direct electrochemical readout?
In a way, any analytical tool that gives you a forest of peaks would be good. That's why Raman or IR would be good. Electrochemical, maybe not, because it just gives a single peak, or a broad peak. So something with a lot of fine structure would be good.
Yeah. Okay, that's clear. Yeah. So do you have
Chris, I have a
still
last
has a question. Yeah.
Okay, I have a last question. Yeah. Okay, really, really great, discussion. I think, maybe I, came back to the very beginning. Wilfred says that, no, not Wilfred. Yeah. Wilhelm says that, during introduction, meant more about the fundings you got, right? Yeah. So yeah, you remember that greatly more fundings you got. So, just a few days we'll get this year's Nobel Prize, right? Yeah, will be announced. So yeah, a question is, for all these wonderful ideas, who wins the Nobel Prize? Is the funding, driven, or the idea driven? Who, be the first. So yeah. Can you give some comments on this? Yeah.
I think it's idea driven in my case, at least.
Okay, it's not a big funding can be, yeah.
Funding is essential, absolutely. But I think we still live in a world where good ideas get funded. you might have to fight hard, but it will be funded. Although, yeah, you would always like it to be easier, but I think it's idea driven, definitely.
Okay, yeah, so because I'm thinking you guys is reservoir computing is from try to get more funding or just you love this.
No, I mean, obviously that's what IBM obviously is interested in exploring how far you can push this. And if you would be able to show that, well, Chris was asking how energy efficient is it? Well, maybe if you can put everything on a chip and have electrical control, and you would say companies would see a reason on making a real device, then the funding becomes very different, but then also the need for funding becomes very different. So if you think about the actual research that we're doing here is relatively simple compared to many other projects that you might be doing.
Yeah, the ideas are the most beautiful things. Okay, congratulations. Yeah.
Thank you.
So, yeah, Chris, that's my question.
Yeah, no, thank you very much, Willem. I think we all thoroughly enjoyed not only your talk, but also the discussion afterwards. And I'm more than happy to hand over the certificate that will be sent to you over email. Yeah, so thank you.
Thank you.
And also all the panelist members very much for today's episode.
Thank you.
Okay, then next week we have Monson Ramani from Nottingham Trent University speaking. And here on this slide, you cannot see that Lan Fu will be the moderator. And in this case, we have that Monson will speak for about half an hour and 2 short talks by Kiani and Takanaka. So looking forward to seeing you all next week again. So that is it for today. Thank you all very much. And just to show who all supports us. Okay, wish you all a very nice weekend ahead. Bye-bye.
Yes.
Bye-bye.
Bye-bye. Very nice talk and discussion. Okay, great.